basically working with DNA, and DNA is the software of our minds and bodies. It designs and essentially controls the design of everything about our biology and that of other species. So like other exponential technologies, and I should say, by the way, I've heard it said that the genomics is the new IT. And the reason they say that is that DNA is actually digital data storage. It's digital. But instead of storing digital data on a silicon chip with ones and zeros, it's storing it biologically in the form of 4 nucleotides that they designate by the letters A, G, C, A. So it's essentially very dense digital storage. And what we do is sequence the DNA in order to decode those strings of software code, essentially. And like other exponential technologies, it is radically, exponentially dropping in price and increasing in volume. So this is sort of an exponential trend that shows the decreasing cost of DNA sequencing. In 1990, $10 per base pair to sequence, and there are about 3 billion base pairs in DNA. And we're now approaching 1/1,000,000th of a cent in 2010, 2011. It took 13 years and $3 billion to sequence the first full human genome. And now it can be done for $10,000 in a month or two, and in the next 5 years, probably $1,000 in a week, within a decade, $100 in a day. So what would that mean if you could sequence your entire genome for essentially the cost and time of a cholesterol test? How might genomics expand to be available and used by many people to drive personalized medicine and lots of other applications? Now, this is a video, watch closely. You can see that, did you? So this is an octopus that has, there are a lot of interesting biological properties. Let me just reverse this, slow motion, so you can see how its texture and color changes to match the seaweed. All right. So why did I show that octopus video? Because it's cool. I'm going to loosely try to make a linkage that as you alter DNA, there are different biological properties that we can. It's mostly cool. So this next image is not a toaster. This is called the Roche GS Junior, Gene Sequencing Junior, and it's essentially literally a desktop gene sequencer. And you can just imagine how much these are getting miniaturized and how much they're decreasing in cost, such that they become accessible to a much larger audience. Maybe you and I have a little Lexmark DNA sequencer on our home offices. But okay, so great, you can sequence DNA, but how helpful is that if you can't do anything with it? Well, there's a thing called 3D printers or synthesizers, and that's the ability to take something that you have that's essentially designed or sequenced, and then output it or print it. Now, of course, that you figure nobody would have access to except large institutions, but I Googled this last night, DNA printers, and I found the following. Here's an ad. Refurbished DNA synthesizer, excellent condition, call for price. So maybe these things will be available on eBay before you know it. I swear this, I did not make this up. This is one of the first Google results. So today, if you can type, you can be a genetic engineer. Now you won't be a particularly good genetic engineer because you don't know what to type yet, but you could be a genetic engineer. And that's really what's driving this level of innovation in genomics. So this is showing a loop of these different technologies because certainly they accelerate on their own, but they also reinforce each other. So as you have advancements in nanotech, it improves your ability to both design data storage and computing as well as do better and more specific genomics. Once you can do better data storage and genomics, then you need artificial intelligence to make sense of this massive amount of data and find patterns. And once you do that, you can find interventions that essentially are at the DNA and genomic level, and then output them with a 3D printer. So you see how they all work together. So let me move on. So putting into practice, how might these things be applied? Let me show you a few examples. On the left is a sugar cube, and on the right is a football stadium. Ralph Merkle, who's one of the leaders in nanotechnology, gave this example at Singularity University that said, if they solve this scale and design issue with nanotech, in the next 10 to 15 years, they should be able to design molecular data storage in 3 dimensions. And if they solve this, it will allow us to store as much data in something the size of a sugar cube as an entire football stadium filled with CD-ROMs.